Good evening to all my biology enthusiasts out there. My dearest children, I have to tell you, you guys make me really happy, and I wish I could do the same as well. I could also make you happy. You know what? The small things that you do makes me really happy. That is, you put down the answers in the comment section. You participate in this whole process of studying. Otherwise, it's just a one-way thing. You get it? I teach and I go and I don't see anything out there. I feel sad. But when you guys participate by answering in the homework sessions out there, by listening to the questions that I put in between the sessions, and then also find the answers, I feel ultimately so proud of you guys. And I want to show the names of those children who did that in the previous session. So we have Prajwal King DC. He said that haploid sperm because meiosis happens in reproductive cells. Homework 1 completed. So the homework 1 I usually give in middle of the session. I just ask a random question and the children, they give the answer. And my dear Prajwal, I'm really happy to see this answer. You also gave the other homework questions answer. Kudos to you. Okay. Now we have Deepika Manohar 6577. Again, my dear child, so good. You have given me the first and the second answer together. Homework 1 says haploid. Homework 2, all the 4 options are correct. That is what you say, and you are absolutely correct. Now we have Saif Sheikh who said D is the answer for H2. So D means that all the 4 options are correct. Again, you have also got the correct answer. Similarly, Saif also commented that 23 chromosomes will be there in the sperm, which is haploid number of chromosomes, my dear children. So again, you guys are doing a wonderful job, and I just wish and pray that I could at least achieve half of what you guys are doing out there. Right. And my dear children, where are we standing now? So we have completed the male and the female reproductive system in a whole. Now we are going ahead with spermatogenesis, oogenesis and its comparison. Initially I planned to do menstruation also in the second part, but menstruation would need another lecture out there. So we would be covering it in the next session, but do not be worried. As of now, we have to cover this and this topic very nicely. They are also equally important. So what in this topic? We will say the steps involved in spermatogenesis, how the sperm formation occur, and then we will see the structure of the sperm, the steps involved in oogenesis, and also we will have a quick comparison between oogenesis and spermatogenesis so that everything settles down in your brain. And if at all you feel that you are not able to catch hold of every single thing out there, it's totally fine to feel that way. Whenever you think that it's totally messed up, pause the video, recall what you have learned, and then go ahead again. Now if you want the notes, what you have to do, you have to be a part of our WhatsApp channel. You get a link there, you can download the notes from there. I will ask the team to put it up if it's not there yet. Okay, and my dear children, you know what, we have given so big help, okay, by just teaching certain kids out there, and they gave us a return gift out there, very big gift, and that is ranks. And they ultimately made us proud the previous year and this year, this year actually, not the previous year. And I want you guys to do the same the next year, and I'm pretty sure you would be able to do that. I'm pretty sure you guys are hard working with the amount of comments I'm seeing out there. It might not be too many, like the 30, 40, no, not like that, but at least 1 to 2 people are there who are continuous with what they are doing. Okay, they are doing it consistently, and I'm telling you, you will beat the other people out there who are in the race with you guys. Okay, well, I wish that you all conquer it together, but yes, this is competition. One has to win, and I'm pretty sure you guys would be the ones who would be winning. So the homework question, the second homework question that I gave was the hymen can be torn by. So the hymen is a cover that partially covers the vagina out there. Okay, so over here, how do you think it can be torn? During the first coitus, that is during the first sexual activity, do you think it can be torn? Yes. Sudden fall or jolt, if you suddenly fall or there is some jolting out there, again it can be torn. Now insertion of vaginal tampon, my dear children, it can happen, it may not break, it may break, but mostly it breaks. Okay, when you insert the vaginal tampon, that is for menstruating ladies, for the blood to be soaked up, you have vaginal tampons, just like how you use sanitary pads. So this is also correct, but in the case of tampon, you have to insert it inside the vagina. Now active participation in cycling and horseback riding, this is also the case where the hymen can be torn. So is the hymen a sign of virginity? No. It's not a sign of virginity because hymen can be torn because of multiple reasons out there. So what is your answer for this particular question? Your answer would be 1, 2, 3 and 4. All the options are correct. Please understand, this is a very important question. Lots and lots of questions comes around this particular concept as well. Okay, so hymen is not an indicator for virginity. And now we are gonna begin spermatogenesis. What do you mean by spermatogenesis, my dear children? We are going to talk about the formation of the little sperms out there. So spermato as in sperm, and genesis means what? Formation of something. So basically we are going to see the steps involved in the formation of the sperm, and it is going to be really fun. So over here we see the fun guys, the fun sperms out there. Okay, let's say a hi and let's begin the session. So I'll tell you a story. You have to remember this in the story format only. Okay, so once Papu hit puberty. Okay, Papu is my friend. Okay, Papu, when he hit the puberty, my dear children, there were slow changes in his body. As in, he had started growing little mustache and also a little beard out there, and there were so many other characteristics that got changed. But within his body, there were much more changes out there. And what were those? Those were related to spermatogenesis, the formation of sperms. So in the case of males, my dear children, spermatogenesis would be starting at the puberty, when a guy hits the puberty. Now at this point of time, there will be a cell. There will be a cell whose name is spermatogonia. Okay? What is its name? Its name is spermatogonia. It's just a normal cell. A normal cell, by that I mean that it has 2n number of chromosomes, that is diploid number of chromosomes. So normal cells of our body has 46 number of chromosomes, right? So here also they have 46 number of chromosomes. How, ma'am? So 2 into n is equals to 46. So n is basically nothing but 23. So 23 chromosomes are there. There is a pair of 23 chromosomes. So 23 basically becomes 23 plus 23 becomes what? 46. Which is why we say that it is diploid in nature. So this spermatogonia which is there thinks that, oh my God, the person has hit puberty. Now I have to start dividing. So it starts dividing. Okay, it ultimately just divides to increase its number. What do you call this division when you just want to increase the number? The number of chromosomes remains the same in the cell, but the number of cells are increasing. So here also it's 2n, here also it's 2n, but it is just dividing. You call this process as mitosis, or you call this as equational division. Why? Because 2n number of chromosomes were there. Here also the cell divided, but it has equal number of chromosomes, just like the parent cell, that is 2n and 2n. So spermatogonia undergoes mitosis to form cells like this. Now what happens? One cell among them thinks that I cannot just keep on dividing. I have to become something in future. I want to become a sperm in future. So it undergoes something that is known as differentiation. What do you mean by differentiation? As in, when you say no, that person has got matured. Or he knows what is correct. He knows that he had to do it this particular way. Similarly over here, this particular cell, okay, got understanding that it has to mature, or basically in scientific terms if we say that it has got matured. There are certain changes in the cell. Okay, and we say that now it has got differentiated or matured, and this process is known as differentiation. And after differentiation, it becomes a whole new person, and you call this guy as the primary spermatocyte. Okay, primary spermatocyte. Ma'am, what happens to the other guy? The other guy will keep on dividing because we need more and more spermatogonia kind of people, right? We need more and more of them. Then only we can get more and more of primary spermatocyte also. You get it? So this undergoes division, but the other one differentiates and forms what? The primary spermatocyte. What is the number of chromosome? Ma'am, the number of chromosome remains the same itself. Now this primary spermatocyte which you got from the spermatogonia, my dear children, what happens to it? It wanted to become the sperm, right? So it tries to do that. Okay, it tries to become the sperm. How? It says that I cannot just undergo mitosis. Okay, I don't think for the formation of sperms I have to undergo mitosis. I have to do something different. I have to undergo something which is known as meiosis. Okay, so it undergoes meiosis, which is a reductional division. What do you mean by that? The number of cells get reduced, ma'am? No. This cell divides into 2 itself, but the number of chromosome becomes half. Okay, so it was 2n. If you half 2n, what it becomes? 2 cancel, it just becomes n. So over here you would have n. Over here also you would have n number of chromosome. So that is why it is known as reductional division, as in the number of chromosome becomes reduced. Now the cells which are formed over here, what do you call it? You call this cell as secondary spermatocyte. Okay, has it become the sperm yet? No, you still have some time. So just to revise, first my dear children, when a person hits puberty, what happens? The spermatogonia which are already there in their body, the male germ cells which are already there in the body, they become active, they undergo mitosis, they just try to increase in number, and then what happens? One cell out there, when every division, my dear children, it undergoes a process of differentiation. Okay, it gets an idea that I have to become something else, and that cell after differentiation, it is not dividing and forming something else, it is just getting differentiated. Okay, it forms something that is known as primary spermatocyte. What is the number of chromosome? It is also diploid. Okay, now the primary spermatocyte says that I want to become the sperm. So it starts another type of division which is known as meiosis. You say it is reductional division. Why? Because the number of chromosome becomes reduced. Now what do you call these type of cells? You call them secondary spermatocyte. But I wanted to become sperm. So again you have to undergo division. So meiosis, whenever it happens, my dear children, always remember it happens in 2 stages. There will be first round of meiosis where you halve the number of chromosome. So this is meiosis 1. There is one more round of meiosis, then only you say that meiosis is completed. Cool. So for that, what we have to do? So don't look at this picture as of now, otherwise you will get confused. So as of now, how many secondary spermatocyte we have? We have 2, ma'am. These are the secondary spermatocyte, right? We got it from the primary spermatocyte. Fine. Now they also undergo meiosis, I told you. This cell undergoes meiosis. This cell also undergoes meiosis. This is the second round of meiosis. Now, ma'am, in meiosis it becomes reduced again, no? No. The second round of meiosis, my dear children, nothing happens. If it was n over here, it remains to be the same. Here also it remains to be the same. So, the primary spermatocyte which is there over here, it's saying that finally we got the sperms, isn't it? But the answer is still a big no. Why? Because these cells which are formed, 1, 2, 3, 4, okay, from one primary spermatocyte, you're getting 4, what? You're getting 4 spermatids. Okay, and these spermatids are not basically sperms. Why? Because they are circular in shape, they are globular in shape. And how does sperm look? They are elongated in shape. So basically there are some structural changes that has to take place. Okay, and that particular changes that is going to happen, my dear children, it would be happening over here, and these spermatids are going to form something that is known as spermatozoa, or you also technically call it as sperms. So if you go in depth, it is not the same, but according to your NCERT, you can just remember this. So spermatids basically form spermatozoa. So they are circular, globular in shape. They slowly become elongated. This process which is there, what do you call that? This is known as Spermatogenesis, as in Spermatogenesis. Okay, you're forming spermatozoa. What do you call the whole process starting from the spermatogonia to this particular thing? You call that as spermatogenesis. That is different. Okay, spermatogenesis as in the whole process. Spermiogenesis as in the globular ones are converting into the elongated ones. So I always remember like this, spermatids, it's round, it sounds like kids, no? So kids are immature, the small kids are there, they are immature, they become mature. So kids become mature. So spermatids become spermatozoa, and this process is known as spermiogenesis. Now the spermatozoa, basically it is all happening where? Inside the testis. And you remember in the previous session I told you, inside the testis what do you have? You have testicular lobules. Inside the testicular lobules you have seminiferous tubules, and inside the seminiferous tubules you have the male germ cell, that is the spermatogonia, and also the Sertoli cell which nourishes this particular guy. So once the spermatozoa is formed, it goes to the Sertoli cells and says, please nourish me, I feel very lonely over here, it's a very new place. So basically the Sertoli cells would be nourishing this guy, that is the spermatozoa. But finally one day spermatozoa has to leave the testis and basically leave the seminiferous tubules and move out. What do you call that particular process as? Where they are finally moving out of the seminiferous tubules out there. You call that process as spermiation. I remember it as aviation. So they are finally moving out of the seminiferous tubule. It is known as spermiation, my dear children. So there are few new terms that you studied. Okay, and I know it's not very easy to remember all of it at once. So take it slow, it's totally fine. Okay, but you have to remember it for a longer time. That should be a motto. Will we be able to remember it for a longer time? Tell me in the comment section right now. Right now, if you think that this particular video is useful, if you think that I am teaching you well, you have to give me one particular answer. That is, do you think you will remember it for a longer time? If yes, you say that, yes ma'am, I'll remember it forever. Can I see that, Josh? I'll be really happy to see that. Okay, so spermatogonia, I'm revising, listen to me carefully. We have spermatogonia. Okay, it undergoes mitosis, 2 cells, 1 cell undergoes differentiation, you get primary spermatocyte. This primary spermatocyte undergoes meiosis because it wants to form the sperm, but meiosis happens in 2 stages. So the first round of meiosis happens, my dear children, you get 2 cells. These are secondary spermatocyte. Okay, now the secondary spermatocyte which is there again undergoes meiosis, you get what? Spermatids. This spermatids undergoes differentiation, again it matures. You have a process to call it, that is known as spermatogenesis. Further, after spermatozoa is formed, it tries to move out, and what do you call this? You call this as spermiation. Okay, so over here if you have a confusion, this is spermatogonia which is dividing itself. Again, my dear children, it will be dividing and forming the, I told you, mitosis happens and further it forms this primary spermatocytes. That's all. And now, one very important thing that you have to remember. Spermatogonia is 2n. Primary spermatocyte which is there, it is also 2n, diploid in nature. Once meiosis starts off, everything becomes what? Haploid in nature. Secondary spermatocyte are haploid, spermatids are haploid, spermatozoa is also haploid. I hope that is clear. Yes, give me a thumbs up, click on the like button if you think this is all useful for you guys. Now, going ahead with what? With the story of the mighty hormones. So everything that I said does not happen naturally, just like that. Basically, there are certain hormones that plays a very important role in this. Okay, and that is why I said that they are what? The mighty hormones. Remember, I told you that in the case of testis, suppose this is the testis that we are having, you have testicular lobules, you have seminiferous tubule, and if I zoom in seminiferous tubule, this is one, this is the other one. Over here you have the germ cell, you have the Sertoli cell. In between the seminiferous tubule, remember you had Leydig cells. And what does this Leydig cell do? It forms, anyone knows the answer out there? It forms the hormone. What hormone? The male sex hormone. And this male sex hormone is the one that will tell the spermatogonia to form the sperm. But who tells this guy to form the hormones? That is also a question, right? So who is the one that says that? I'll tell you. So in our body, in our brain, we have a region which is known as hypothalamus. Okay, and hypothalamus releases a hormone which is known as GnRH. That is gonadotropin releasing hormones. Okay, hypothalamus, which is a part of our brain, releases GnRH, which is gonadotropin releasing hormones. Okay, now this gonadotropin releasing hormones which is there, it will be going and acting on what? On the pituitary gland. That is the next gland out there. Pituitary gland. Where is the pituitary gland? Again in our brain itself. So this guy goes and acts on the pituitary gland and basically releases what? Gonadotropin, right? It is gonadotropin releasing hormones. So basically this guy is going over here and telling to release what? Gonadotropins. What are the gonadotropins released by the pituitary gland? It is FSH and LH. It is FSH and LH. Listen to me. Hypothalamus releases a hormone, and this hormone goes and acts on the pituitary gland. Pituitary gland releases two hormones. One is FSH and the other one is LH. FSH stands for follicle stimulating hormone, and LH stands for luteinizing. The spelling, I always get it wrong. Where is it? Is it there? Oh my God, it's not there. Okay. Okay, children, please check the spelling. I am really bad. Remember, I am also a human being. So, not I am bad, my spelling skills are bad. So, it is, I suppose, luteinizing hormone. Anyways, we call it LH. Usually, we call it LH. So, that is how I remember it. So, we have this hormone and this hormone, right? Now, what do these guys do in the case of sperm formation? How do they help? So, the story begins over here. So, this is the seminiferous tubule and this is the outer space. So, here you have the spermatogonia. Okay, this is the spermatogonia. Here you have the, what do you call, Sertoli cells. Sertoli cells. Okay, so this guy has to form the sperms. That is why I wrote it like that. Spermatogonia forms the sperm. Here you have the Sertoli cells. Within what? The seminiferous tubule. Now outside it, what do you have? You have the Leydig cell. Leydig cell, right? So the pituitary gland releases two hormones and the LH is going to act on the Leydig cell. The LH, you can remember like that. LH acts on the Leydig cell and makes it releases what? Androgen, that is the male sex hormone. Now, what is the most famous male sex hormone? It is testosterone. Testosterone. Okay, now this testosterone, my dear children, will be going over here and helping in the formation of sperms. But ma'am, pituitary gland also releases one more thing, that is FSH. What does that do? So, follicle stimulating hormone, my dear children, it will be acting on the next cell, which is Sertoli cell. It says that, hey, can't you see the testosterone, everything is being formed? You have a sole responsibility for nourishment, right? What are you doing over here without any work? You have to release a very important thing, which is known as ABP or androgen. Can you see it? Androgen binding protein. It's not there in your NCERT. Just understand. Okay. So in your NCERT it is said that FSH acts on Sertoli cells and Sertoli cells releases some factors. Now you won't be able to remember it if I say it releases some factors. So please remember the story. So this guy LH went to Leydig cell and said that please release androgen. Right. This androgen went inside the seminiferous tubule and is helping in this particular process. But these guys, the androgens which are there, they are like, we'll come and we'll go to the pub and we'll just come back over here after some time. But there should be someone to stop them because they have work to do. Right, they have to help in the formation of sperm. So there should be someone who could bind all this testosterone or the androgens together over here at this particular point of time. Right, and who is that particular thing, or what is that particular thing which would be helping in binding? It is the ABP. So follicle stimulating hormone tells the Sertoli cell that, hey, see, testosterone will go now, you have to bind it by producing androgen binding protein. So androgen binding protein helps in making the testosterone stay here itself and help in the formation of sperm. Specifically in your NCERT, one line is given. It acts on Sertoli cells, stimulating secretion of factors, helping in spermatogenesis. So the most crucial role is played over here during spermatogenesis. That is, you remember I told you, you have the, where is it? I told you, you have the spermatids, which are forming spermatozoa. right? And this process is known as spermiogenesis. So majorly during this particular process, you would be needing lots and lots of testosterone. Anyway, you would be needing testosterone during the whole process, but here you need it better, okay, or it's the most crucial thing out there. I hope now you get what I'm trying to tell you. Now to sum this up, again I'll review everything so that you understand it better. So first thing first, spermatogenesis starts at puberty. Why? That should have been a question from your end, okay, but you cannot obviously question me because this is not a live session, but again I can tell you. So spermatogenesis starts at puberty because the hypothalamus right over here would be releasing what? GnRH, okay. Now what happens? This gonadotropin releasing hormone, my dear children, would be acting on the pituitary to release two more hormones, FSH and LH. The LH will go and act on the Leydig cell to release androgens. The androgen comes inside and helps in spermatogenesis, but they feel very bored, they try to move out. Okay, but then who is there to stop? It is the nurse cells, the one who takes care of them, the Sertoli cells, right? So FSH say Sertoli cells, be more careful, release ABP, so that basically all the testosterone remains here itself and help in the process of spermiogenesis. I hope that's clear. Okay, so someone came in between, so I got lost a bit. So I hope I said the initial part correctly. Spermatogenesis occurs, starts with puberty. Why? Because, my dear children, hypothalamus starts releasing GnRH at that particular point of time, because there are certain changes in our body that is happening. Done? So this is the story of mighty hormones. Did you understand? Did you understand? I hope you understand. How do I understand that whether you understood or not? By your comments, by your likes, by you telling me, ma'am, I am not understanding this particular thing. Can you explain it again? So I will be seeing those comments as well. I will be seeing the good comments, the bad comments, everything is visible to me, right? And I will be responding to them. Okay, so please don't worry. If you have any doubts, put it down there. Cool. Now going ahead, I hope the spermatogenesis part is clear, and if at all you have doubt in spermatogenesis still there, when we will be comparing between spermatogenesis and oogenesis, all of it will be settled down. If you think it's too much for you, I'm telling you again, please pause the video and try it once more. Okay, it will be helpful. Now, structure of the sperm, the main character, right, the main guy out there. So say hi to the survivor. That is what I have written. Why have I written this? Because you know what, sperm, when it's entered the female body thinks that it's something foreign and would try to kill the sperm by it's very acidic inside the female body, inside the vaginal area, my dear children, so it will try to kill it. But still, some sperm would be able to survive, and survive until how many days? 5 days. Okay, so please understand that. Please have that little cute, fun facts about all these things. So that is why I called the sperm the survivor. Now going ahead with its structure. So my dear children, okay, so over here I'll just roughly make something. Okay, you do not have to follow me, you have to follow this, but just for your understanding. So you have a head region in the case of sperm. Okay, and you have a neck region. Then you have a middle region. So if it's a middle region, it's in middle of what? It's in middle of the tail which is there, and also the head which is there. So this is the head, this is the neck, this is the middle piece, and this is the tail. And what is inside all of this thing? So inside the head you have, this is a cell, right? Sperm is a cell, and cell will have nucleus. So this also has an haploid nucleus. Okay, as all of you said, Prajwal said, Deepika said, no, Saif said, and let me see the name. I have to tell. Yes, Deepika itself. It was correct. Yes. So where was I? Deepika said that all of it, they are haploid in nature. They have half the number of chromosomes. The sperm contains half, the egg contains half, both of them come together, half plus half, 1. So basically it becomes diploid in nature. N and N, 2N. Okay, so over here it has the head region which is having the haploid nucleus, and then there is neck. And over here in the middle piece, my dear children, you have strands of mitochondria. You would have multiple mitochondria over here. Why, ma'am? Because this mitochondria is the one that will provide energy for the movement of the tail. And if at all tail is not moving, do you think it can reach the egg, which is where? Where is the egg located in the case of Where is the egg located in the case of females? Where in the reproductive tract? Homework number 1. I taught you in the previous session, now you have to give me the answer. Okay, so yeah, that is the one that helps in the movement of the tail. Now, is that all, ma'am? No. One very important thing, the whole body of the sperm is covered by something that is known as the plasma membrane, and that's not all. At the head of the sperm, at the top of it, you have something like this, like a crown, because it's a survivor, right? What is this thing? That is a shield actually, not the crown, it's a shield. Okay, why this shield? What does it have and what do you call it? So this shield which is there, it is known as acrosome. And what is acrosome? Acrosome is nothing but a enzyme filled sac out there. Okay, it's an enzyme filled sac out there. What enzymes, ma'am? Basically, when the sperm and the egg comes in contact, the egg will have multiple coverings around it. So there should be something to dilute all these coverings and get into the sperm, to break down all the barriers and get to meet its lover out there, that is the egg out there. You get it? This is Laila Majnun story out here that is happening. So acrosome contains what? Enzymes. You call them hydrolytic enzymes. It is going to break all the hydrogen bonds out there. Hydrolytic enzymes are there over here in the acrosome region. Okay, so now we will study the structure of sperm. So over here, as you can see, head, neck, middle piece and tail is there. Head contains haploid nucleus and also the acrosome. What is acrosome? Acrosomes contains enzymes for fertilization process. Then you have middle piece that contains mitochondria for what? Energy for the movement of the tail. Tail does what? It enables sperm motility. That means it helps in the motility or the movement of the sperm. Done, which is very essential for the process of fertilization. That's all. So over here you have a picture of it as well. Let's see. So this is the acrosome, this is the head region with the nucleus containing the chromosomal material, and there is a neck region, there is middle piece with the mitochondria and the tail, my dear children, and over here you can see the whole body is covered by the covered by the plasma membrane, including the acrosome out there. Cool? Okay? And that's how we completed spermatogenesis and the structure of sperm as well. And now we are going ahead with the next process, which is known as oogenesis. Okay, are we ready? Are we ready for that? So I hope everything is settled in from the other part. Now we are going ahead with deeper sense. We are going to talk about the journey of oogonia. So journey of spermatogonia, if you remember, it started at puberty. Until then it was just waiting. One day it will happen, one day I will also form sperm. But in the case of oogonia, it does not have time to rest. Okay, so when the baby is born, suppose it's a girl baby will have ovary, right? Fetal ovary. The fetus will have ovary, which is why we call it fetal ovary. In the fetal ovary itself, the formation of oogonia occurs. The oogonia is already there. Oogonia is what? The female germ mother cell. Okay, that is oogonia, the gamete mother cell out there. And this gamete mother cell, it's not just present. It's not just present, it starts dividing. So spermatogonia is also present, no, from starting point. But oogonia, my dear children, starts its division during what? The embryonic development. When the baby is not actually even a baby, it's just undergoing the embryonic development. At this particular point of time, the oogonia which is there are so many oogonia. How many? Millions. Oh my dear children, just a second, okay. I just went ahead. I have to tell you one very important thing. I'm so sorry. Please come back again. Please come back again. So, ejaculation. In the case of sperm, there are 200 to 300 million sperms being released. Okay, since we spoke about million there, I thought of it now. So, 200 to how many? 300 million sperms are being released. And out of it, please remember, 60% should be normal shape and size and 40% motile for fertility. Remember, very important. How much percentage with normal shape and size? 60. Okay, 60 of them should be having normal shape and size, and also 40%. Now, out of that, 40% of that should be at least having good motility as well. It should have nice movement as well. Please remember this. You get questions from this part as well. Okay, now we go back. So oogonia, the journey of oogonia. So we are in the embryonic fetal stage, my dear children. Over there are millions of oogonia out there and they have started division. Remember, no new oogonia are added after birth. After birth, there is no addition of new oogonia there. Whatever oogonia is there, it is undergoing division. It also undergoes mitosis, increases its number just like how spermatogonia did, right? Now what happens is that oogonia, my dear children, it says that that's all. I do not want to go ahead with anything. I actually want to form the eggs out there. So let's start the division. So oogonia enters meiosis. Okay. The oogonia guy which is there, not the guy, the girl, okay. I usually say everyone guy. So oogonia which is there, which is having 2n number of chromosome, it says that let's start meiotic division. It undergoes, it increases its number and things like that. It also differentiates and finally it starts off meiosis. You know, meiosis happens in multiple stages. Meiosis means you have prophase, you have metaphase, you have anaphase, and finally you have something that is known as telophase. That is when you say that one round of meiosis have occurred. Now in the second round, prophase 2, metaphase 2, anaphase 2 and telophase 2. When that occurs completely, you say meiosis is completed. So this oogonia which is there, it undergoes division, okay, it undergoes mitotic division. Later it thinks that I have to start off with meiosis. So it starts off with meiosis, my dear children, and over here it gets arrested. Arrested as in it is not able to move forward, it is not able to complete the meiosis 1 division. It is stuck in prophase 1, and in the stuck stage where it is not anymore oogonia, it has lost that tag. It is now known as what? The primary oocyte. It has just started off the division, it has not even completed meiosis 1, it is stuck there in prophase 1 and you call this as the primary oocyte. What is the number of chromosome? Has it completed meiosis? No, which is why the number of chromosome remains to be 2n itself. There is no change. Now what happens? Each primary oocyte is surrounded by granulosa cells forming primary follicle. So the poor oogonia wanted to form the ovum, but that is not happening. And later what happened? So many guys came in and said that no, we are not going to allow to start division out there. Okay, we are going to form a barrier around it. So basically over here the primary oocyte, remember I am telling very important things out there. The primary oocyte are surrounded by so many cells out there. You call them granulosa cells. Or let's change the story, okay, or let's change the story. So over here you got oogonia, the oogonia started division, it underwent until prophase 1 and then it got stuck. So it was so sad, okay, it became so sad, so devastated. So what happened, all our, remember our grannies, how are they, grandmothers, they're all very cute, no, they pamper us a lot, okay. I lost my grandmother at a very young stage, but I still remember her. So she was very cute, okay. So similarly over here, all these grannies came together to pamper them. So these cells are known as granulosa cells out there. So it comes together and this whole structure now will be known as the primary follicle. Okay, granulosa cell plus the primary oocyte is known as the primary follicle. So this thing has formed. Now what happens? Many follicles degenerate from birth to puberty. You know what? Still it is not forming the egg. From that particular stage, from birth, where it has formed the primary follicle, it is now, my dear children, millions of them, they formed this and you have lost so many primary follicles, so many of them just gave up. Okay, the grannies also gave up, the primary oocytes also gave up, my dear children. How many primary follicle will you have at the end? Only 60,000 to 80,000 primary follicles per ovary. So in this ovary 60 to 80,000, and this also 60 to 80,000. Only that much would be left in each ovary. Now one fine day, what happens? So the primary follicles would soon slowly, when you hit puberty, my dear children, okay, when we hit puberty, one fine day the primary follicle felt something different out there. The primary follicle again was surrounded by new sets of granulosa cells. New sets of granulosa cells, and also one more layer was formed around it. You know, when we are very sad, you get multiple people on there to tell that it's okay, it's okay. Similarly, over here, primary follicle were very sad. It reached puberty. Almost so many lost their lives. Only 60 to 80,000 are left. Now, at this point of time, what happened? More granulosa cells came together. Remember, more granulosa cells came together, and apart from that, a theca layer, a new theca, T-H-E-C-A, T-H-E-C-A, theca layer is being formed over here around this particular thing. So now can you call it primary follicle anymore? No. You will call this new structure as the secondary follicle. But inside the secondary follicle, it still remains the same. It's still the primary oocyte itself. Nothing has changed there. The primary oocyte remains the same. The follicle, if you see, the name has changed to what? Secondary follicle. This happens when? During the puberty, my dear children. More layers of granulosa cells and the theca layer formation occurs over here. So we'll just revise. First we have the oogonia, my dear children. In the fetal stage itself, they undergo mitosis, they increase their number. Later they start meiosis and they get stuck in prophase 1 of meiosis 1, my dear children, which is why you will call them, you will rename them primary oocyte. It felt so bad, so grannies come together, granulosa cells, perhaps primary oocyte becomes primary follicle. It stays like that until puberty, and so many of them are lost. Only 60 to 80,000 of them are left. And then to encourage them, what do you have? You have more granulosa cells and also one theca layer. And this is now known as secondary follicle. Secondary follicle still has the primary oocyte, which is still stuck in prophase 1, and the ploidy is what? Still 2n. Now what happens to the secondary follicle? Let's see that. So the great switch from secondary follicle to what? Tertiary follicle. We are gonna talk about that. So as of now, you have the primary oocyte, okay, the granulosa cells, and the next granulosa cells which came in, okay, and one extra, okay, it cannot be visible, so I will make it like this. One extra layer that has come in, that is theca. So this is what? This is the secondary follicle. Now my dear children, what happens is that each time when you have ovulation at this particular point of time, again certain hormones would be coming in. Okay, certain hormones would be coming in. So in puberty, FSH, follicle stimulating hormone, it's not just in males, in the case of females also it's there. It is the one that says that primary follicle you have to make secondary follicle. Hey, secondary follicle, you would have to make the tertiary follicle. Then again and again it has to do everything fast, because at the end what do we want? We want the ovum, because each month the female has to release one ovum, expecting that the sperm would be coming, fertilizing, and then you can have a baby. Okay, so this process has to happen. So every time, my dear children, the follicles keeps on generating like this. So over here the secondary follicle will undergo certain changes, as in what changes? So there will be more granulosa cell, that is there. Apart from that, my dear children, I will make the changes here itself. Okay, here itself I am going to make the change. The theca layer which is there, it forms into two theca layers. It was one, right? It is now going to form two theca layers. The one inside is known as inner theca and the one outside is known as outer theca. Okay, and not just that, there will be a cavity being formed over here. There will be a cavity being formed over here. What do you call this cavity? You call this cavity as antrum. Well, why all this thing is happening? And what do you call this now? This is not secondary follicle anymore. You will call this particular thing as the tertiary follicle. And the question, why is this happening? Why there is inner theca interna? Why there is outer theca externa? Why there is antrum? So basically, the tertiary follicle which is there it is preparing for a new thing out there. That is the primary oocyte. It is preparing for welcoming the secondary oocyte. Where do you get the secondary oocyte from? From this guy, that is the primary oocyte. Remember, it was stuck in a stage. It was stuck in prophase 1 of meiosis 1. It finally undergoes division. You get 2 cells. Okay. One cell which is there, it is known as the polar body. The first polar body. The second cell which is there, it is known as the secondary oocyte. And now once it has completed meiosis 1, it will reduce its ploidy. It is N and N over here. So over here, this guy, we have made a swimming pool, we have made boundaries, everything for what? For this secondary oocyte. So antrum is nothing but the swimming pool. You can remember like that. I don't have any problem if you remember like that. You just have to remember it. Okay. So over here, it forms the secondary oocyte and also the polar body, the first polar body. My dear children, remember, the first polar body cannot undergo any more division. Okay. There is only one. After so long, there is a child being born. There is only one child being born. Okay, there is no 2 child. The second child is not able to make it up. Okay, so the first polar body remains like that. Okay, degenerates slowly. The secondary oocyte, it remains the same. In the case of sperm, we saw that both of them divides. You get 2 cells. This also divides. You get 2 cells. Here also you get 2 cells. But here, what happened? This division didn't take place properly. This guy is not able to divide. So we cannot expect this to happen. Will this happen now? The secondary oocyte, do you think it can give rise to 2 ovum out there? Let's see that now. So I hope that's clear. So secondary follicle, what does it have? It has granulosa cell and a theca layer. In the case of tertiary follicle, you have granulosa cells and also inner theca interna, outer theca externa, and also one swimming pool, that is the antrum. And finally, within the tertiary follicle, what happens? The primary oocyte divides into secondary oocyte and the first polar body. Done? Okay, why does this happen? Why is not able to form fully? Because it wants that all the cytoplasm and everything which is there is utilized by this guy, because this guy has the potential to form the ovum later on. Okay, now that has happened, my dear children. Now what? Now finally comes in the ovum. Let's see that. So graphene follicle development and ovulation. Now the tertiary follicle which is there, it will form graphene follicle. So the story is not yet complete. Okay. So over here in the case of graphene follicle, what's different? Again, FSH, follicle stimulating hormone, is saying tertiary follicle, you cannot remain the same, you have to form something else. Okay, the baby is growing, you have to do something. So over here, the baby as in I'm talking about secondary side, not the actual baby. Okay. So over here in the case of graphene follicle, my dear children, what did you have? You had the secondary oocyte, right? With half the number of chromosome, n number of chromosome, because it is the result of meiosis 1. Now over here around it, what do you have? You have the granulosa cells. Agreed? And you have antrum. Agreed? So all of it is there. Okay, this portion is the antrum. And then, my dear children, you have theca, internal one, the external one, everything is there. What is just like the tertiary follicle, what is extra about the graphene follicle? The extra thing about the graphene follicle is that this secondary oocyte which is there, it will undergo division? No, it does not undergo division. Instead, it will form a new layer around it itself. It is now self-grown. It says that I can manage myself. Okay, so that's happened that when kids grow up. So basically over here, it forms its own layer. It is made by secondary oocyte. Please remember, it's very, very important. It is known as the zona pellucida layer. Now this is for what? This is for its own protection out there. Okay, it has zona pellucida layer, and apart from that, the granulosa cells over here, they also form an extra thing over here. Okay, that is done by granulosa cells, not by the secondary oocyte. And this extra thing which is there, you call this as, where is it? You call this extra thing as corona radiata. Corona radiata. So basically secondary oocyte which is there, it has two things around it. One is the zona pellucida, and the second thing that you have is the corona radiata. Zona pellucida is something that it itself makes, and seeing that the granules become happy and they form what? Corona radiata for itself, for them, for the secondary oocyte as well. Now my dear children, finally comes in what? The ovum still does not form. Okay, always remember secondary oocyte also tries to undergo meiosis 2. It also begins the meiosis 2 process, but it gets stuck. Okay, and in which phase? This is homework 2 for you. So today you have 3 homeworks in total. The first one I already told you, this is the second one. Okay, in what stage of meiosis 2 does this particular secondary oocyte get stuck? It is not able to form the ovum. When does it form the ovum? When actually the sperm would be coming in. When the sperm and the egg comes in, my dear children, sperm and the secondary oocyte comes in. This secondary oocyte, when the sperm is very near, very close to it, when it comes and attaches, my dear children, slowly undergoes division to form what? The ovum out there. Okay, and 2 ovum? No. Again, the secondary oocyte which is there, it also would not be able to do a division like this. So there will be one small cell that is known as the polar body out there, second polar body. So this was the first polar body, this is the second polar body out there, and here you get the ovum at the end, but this is during fertilization. Okay, always remember that. Okay, done. Now, a comparison between spermatogenesis and oogenesis, with which you will understand everything if you have not understood. We will begin with oogenesis first, because we have just studied it. We need to have a recap of it. Okay, so first thing that happens is oogonia is formed. Oogonia in the fetal ovary itself, it undergoes mitosis and then it undergoes differentiation as well. Okay, finally, my dear children, it undergoes a process that is known as meiosis. But is it able to complete it? No. It gets stuck in a phase that is known as prophase 1. What do you call this? Prophase 1. And at this point of time, you will have a different name for it. It is known as primary oocyte. Now, the primary oocyte which is there, my dear children, what will happen to it? The primary oocyte which is being formed over here, what will happen to it? They have actually put down the names so top, you do not understand what they are trying to say. So this is the primary oocyte which is being formed. Okay, and the primary oocyte will be covered by so many granular cell layers. Okay, and it is known as primary follicle now. Now slowly at the puberty, my dear children, what will happen? So many follicles degenerate, only few will be left, and then it feels so sad. So to support it, you have secondary follicle being formed. That is, more granular cells comes together, and also you get one extra theca layer around it. So that is the secondary follicle which is being formed over there. Now what happens, my dear children? Secondary follicle will soon transform into tertiary follicle. Okay. In tertiary follicle, the theca layer differentiates into 2 layers, okay, and there is an antrum formation. And finally, the primary oocyte divides into secondary oocyte and also the first polar body. Okay, done. So this happens after puberty. Okay, after puberty, the secondary oocyte and everything is being, secondary follicles and the secondary oocyte and everything is being formed. So secondary oocyte is being formed, the first polar body is being formed, and then what happens? The secondary oocyte which is there inside the tertiary follicle remains the same, but the follicles now convert into what? Graphian follicle. It matures into Graphian follicle. And later what happens, my dear children? When it matures into Graphian follicle, basically the secondary oocyte which is there, it releases something around it that is known as, it forms something around it that is known as zona pellucida. And seeing that, granular cells also would be making corona radiata. So secondary oocyte is surrounded by zona pellucida and also corona radiata. And finally, when fertilization will happen at that particular point of time, okay, before fertilization happens, okay, what will happen? The secondary oocyte will complete its division to give ovum and also the second polar body. Okay, so over here, if you are asked about the secondary oocyte, it will be having 2n number of chromosomes. Okay, when it forms the primary oocyte, it will be having 2n number of chromosome. When it forms the secondary oocyte, it will be half, that is n. Here also it remains the same, that is n. So from one primary oocyte, Okay, you are getting just one ovum out there. But here in the case of males, my dear children, what is the case? Here from one primary spermatocyte, it undergoes division, forms secondary spermatocyte. This also undergoes division. You get 2, 2, 2, 2 here. That means 4 sperms are being formed. That is one big difference between spermatogenesis and oogenesis. So if at all in the question it is given that there are 100 spermatogonia out there, okay, or basically 100 primary spermatocytes out there, how many sperms will be formed? 100 into 4, it will be 400 sperms that will be formed. Over here the question is, if there are 100 primary oocyte, how many ovum will be formed? It will be just one, that is 100. 100 into 1 is basically what? 100 itself. I hope that is clear. Okay, now that can be a little confusing, but it's totally fine. I told you, it's okay. Revisit, re-revise, put it in 2x speed and listen to it once more. Totally fine, but you will get it. Or you can read your NCERT also. In that way also, it will be sorted in your head. Okay, cool. Can we do the questions? I hope we can do the questions, starting with the first one. The immature diploid male germ cell that produces sperm are what? So first thing, you have to look for the term diploid. Okay, 2n number of chromosome. So spermatogonia, does it have 2n number of chromosome? Yes, ma'am. It has not undergone meiosis, no, so it has 2n number of chromosome. Secondary spermatocyte? No. No, ma'am. Why? Because primary spermatocyte undergoes meiosis 1, and that's how you get secondary spermatocyte. So this guy has 2n, this has n and n, so it is haploid. So this is wrong. Now spermatids, ma'am? No. Why? Because this also again undergoes division, and that's how it forms spermatids. This has n number of chromosome itself. So this is also haploid, not diploid. Wrong. Spermatozoa, ma'am, this guy matures, undergoes differentiation to form this. So number of chromosome does not change, it remains n only, it is haploid. So what is diploid? Only this guy is diploid over here. Okay, so what is your answer? Your answer becomes A. Now the next question now there, I hope you got it. Okay, now which of the following is correct with respect to the sperms of the fertile males? At least 40% of sperms must have a normal shape and size. Children, if you know the answer first, you can tell me. Okay, now at least 60% of the sperms must have motility. At least 60% of sperms must have normal shape and size, both A and B. What is the answer? Is it both A and B? Because ma'am, we saw 60 and 40. But my dear children, there in the book, 60% was for what? So 60% were the ones that have normal shape and size, and 40% were the one that has the motility. Right? So here opposite is given. So is it correct? No. So what is the only thing that is correct? The answer to this question is C, which is how you lose marks without reading it properly. Cool. Now the next question. The process of spermatogenesis and oogenesis is started respectively at what point spermatogenesis, formation of sperms, and oogenesis starts? So you know spermatogenesis hits during puberty. Oogenesis happens during embryonic stage itself. So puberty and menopause, embryonic stage and menarche, puberty and embryonic stage. Your answer would be puberty and embryonic stage, even though at this point of time you do not know what is menopause and menarche, or probably you might know, you might have studied in school, but still I am telling you. Okay, so your answer is this. Now fourth question, the first meiotic division during oogenesis is completed at the stage of, remember the question, read the question precisely, okay. The first meiotic division during oogenesis is completed at the stage of, when does the first division complete? When does the primary oocyte finally get the swimming pool? In the tertiary follicle, right, in the tertiary follicle. So my dear children, primary oocyte within the primary follicle? No. Primary oocyte within the secondary follicle? No. Primary oocyte within the tertiary follicle. That is where it will undergo first meiotic division. So children get confused with this and this. Secondary oocyte within tertiary follicle. So it is not said what is formed after the first meiotic division. So the first meiotic division during oogenesis is completed at the stage of what? The primary oocyte within the tertiary follicle. This has to undergo division. Then only we will say completed. Okay, so this won't be the correct answer. This is the one that would be your answer. I know that's a bit tricky, but yes. Okay, and now we have homework, and let's see who are going to be the winners in the next session. Okay, so the ploidy of spermatogonia as in primary spermatocyte, secondary spermatocyte and spermatid is. So whoever attended this class carefully will give the answer. So it's asking whether it's diploid, it's haploid, what is haploid, which one is diploid and things like that. Okay, so I hope you answer that, and we will be seeing in the next session with menstruation. My dear children, I will be teaching that very nicely so that you all understand. If at all you still think that I teach nicely, you are able to understand things and you can trust me, well, you have to trust me. Why? Because of this. Okay, so you have to trust me because of this. But okay, you can take your time to trust, and my dear children, please put your valuable feedbacks down in the comment section, and please recommend it to your friends if at all you feel it's helpful. Wishing you good luck, lots and lots of love from Jasmine Ma'am. Bye bye, take care.